Hi students, once more welcome back to these video lectures of the fourth sem Chemistry for Biological Sciences paper, the chapter Nucleic Acid. The first part of this first part of this Nucleic Acid chapter, a video lecture is uploaded in your Google Classroom 3 or 4 months ago. So here I am going to restart this video lecture about the nucleic acid. Already in our previous lecture or in our Google Meet class, we discussed about what is nucleic acid. It contains nucleoside or nucleotide, and nucleotide is the it contains the pentose sugar, heterocyclic nitrogenous base and phosphate group. What are the pentose sugar that is present in nucleoside is that is ribose or 2-deoxyribose. It is known as 2-deoxyribose because of the absence of oxygen group in second position of the carbon. And what are the heterocyclic nitrogenous base present in nucleic acids are the substituted of purine and pyrimidine bases, and that are purine bases are adenine and guanine, and pyrimidine bases are cytosine, thiamine and uracil. And how this nucleotide with this heterocyclic nitrogenous base, that means the pentose sugar and heterocyclic nitrogenous base are linked together. Already we saw in a nucleotide in the previous lecture or class. So here we are going to discuss in detail about the what is nucleic acid structure. Nucleic acid is nothing but a biopolymer. It is the polynucleotide. That means nucleotide is linked together to form a polymer. So it is known as a polynucleotide and having very high molecular masses. And these nucleotides are linked through by a phosphate ester linkage. This nucleic acid contain pentose sugar, heterocyclic nitrogenous base and a phosphate group. And it is a biopolymer of cell responsible for the biosynthesis of protein, and important biological function of this nucleic acid is the transmission of hereditary character. Depending upon the pentose sugar which is present in nucleic acid, it can be divided into two types. One is ribonucleic acid. In that, the pentose sugar present is ribose, that is known as RNA, and second one is deoxyribonucleic acid, DNA. In this, the pentose sugar present is 2-deoxyribose. And heterogeneous bases present in nucleic acid in the case of RNA are adenine, guanine, cytosine and uracil. Here the thymine is not present in RNA. The nitrogenous bases that are found in DNA are adenine, guanine, cytosine and thymine. Uracil is absent in the case of DNA. So the difference between this RNA and DNA is the nitrogenous base thymine is present in the case of DNA instead of uracil that is present in RNA. So this phosphate, this nucleotide is linked together through this phosphodiester bond, that is by one pentose sugar, fifth carbon is linked to the third carbon of the another pentose sugar through this phosphodiester bond. This is the primary structure of this nucleic acid. One pentose sugar unit, one base, and this pentose sugar unit is linked through a phosphodiester bond. Next is the structure of RNA. RNA, we already see that this polynucleotide chain are linked through phosphate ester linkage, and pentose sugar present in here is D-ribose. And the nitrogenous bases are adenine, guanine, cytosine and uracil. And this phosphate linkage is through the C5' of 1 ribose through the to the C3 dash of the next ribose unit. And this RNA is mainly present as a single-stranded structure. Sometime what will happen, this single-stranded structure may fall back and itself like a hairpin in some section, and it will form a double helical like structure. So that double helical like structure is formed by base pairing of principle. That means the adenine will pair with the uracil while guanine will pair with the cytosine. There are mainly 3 types of RNA present in cell. These are messenger RNA, transfer RNA and ribosomal RNA. What is messenger RNA, or it is known as template RNA? Messenger RNA is a single-stranded molecule. It is a base sequence complementary to parental DNA template strand on which it is synthesized. So that is why it is known as template RNA. Although it strand may exist with some degree of coiling. A single-stranded molecule, sometime it will undergo coiling, and but this coiling means it will not forming a double helical like structure through this base pairing. So it is the most heterogeneous in size and stability among these 3 types of RNA, and it has a large molecular masses at about 30,000 to 50,000 molecular masses, and found either free in cytoplasm or associated with ribosomes. It contains 5% of total cellular RNA. It constitutes about 5% of total cellular RNA. Main biological function of this messenger RNA is that it carries genetic information or message from chromosomal DNA to ribosomes for protein synthesis. That protein synthesis part we are going to see in the next videos, detailed next video. Second one is the transfer RNA or soluble RNA. It is also a single-stranded molecule. Sometimes this strand bending and looping by itself in a clover leaf pattern. It is the smallest polymeric form of this RNA with a molecular mass of about 25,000 and contains about 70 to 100 nucleotide unit. It occurs in a soluble cytoplasm and constitute about 50% of its total cellular RNA. The most important biological function of this transfer RNA is to transfer or carry specific activated amino acid from the amino acid pool of the cytoplasm to the correct site on the ribosomes during protein synthesis. But also we can see there in the protein synthesis part. And next one is the ribosomal RNA. Ribosomal RNA is a single-stranded molecule with a high degree of coiling. And due to this high degree of coiling, double helical region is more in the case of ribosomal RNA. And this has the highest molecular mass of around 10 raised to 6 molecular mass among the 3 types of RNA, and also it is also most abundant one and makes up about 80% of the total cellular RNA. It is present in the ribosomes. In its biological function, it serves as the major structural component of ribosomes. The exact biological function is not fully understood. Believed that it provides a correct orientation to the messenger RNA, so that this transfer RNA molecule will able to translate message on them into the synthesis of specific protein. Thank you. In the next video, we will discuss about the double helical structure of DNA.